You know, when we look at pharmacology, I think there's this really strong tendency to just view antibiotics as like simple off switches for infection. Take the pill, the bacteria die at the end. But looking through this stack of pharmacology notes on our desk today, we're doing a deep dive into quinolones, folic acid antagonists, and urinary tract antiseptics for you. And it reveals this incredibly complex, like highly specific battleground.
It really does. It's not just a blunt instrument.
Right. We're looking at how these drugs actually function at the molecular level. And it turns out treating an infection is it's way less about dropping a bomb and much more about exploiting these tiny microscopic vulnerabilities.
Yeah, and the fundamental goal here, the concept that really underpins all these drugs, is something called selective toxicity.
Okay.
So we are looking for very specific targets, whether that's a metabolic pathway or a structural protein or maybe an enzyme.
Something the bacteria needs.
Exactly. Something absolutely vital for the bacterial survival, but, and this is the key, entirely absent or at least structurally very different in human biology.
Because you don't want to hurt the patient.
Right. You want pharmacological precision. You want to neutralize the pathogen, without wrecking the patient's own cellular machinery in the process.
Which is such a delicate balance, right? And as we go through these sources with you today, you'll see how brilliant that precision can be. I mean, we're going to talk about drugs that drop literal formaldehyde directly into a bladder infection.
That is a wild mechanism.
It's mind-blowing, but we also see how messy the collateral damage can get. Like, we'll get into why a simple glass of milk can completely disarm a super potent antibiotic.
Yes, the chelation issue.
Yeah, and how just tweaking a drug's chemistry slightly expands its reach to totally new families of bacteria. But let's start by looking at how we attack a bacterium's ability to manage its own genetic code.
Okay, so to understand this first major class, the fluoroquinolones, we have to talk about bacterial DNA. Because bacterial chromosomes are massive. Like, compared to the physical size of the cell itself, they are huge.
So they have to cram it all in there.
Exactly.
Yeah.
To fit inside, the DNA has to be super coiled. It is wound up under this immense torsional strain.
Okay, I was visualizing this when I read the notes. It's like a hopelessly tangled pair of wired headphones, or like a garden hose twisted in on itself.
That's a perfect analogy. And the problem for the bacteria is you can't actually read the genetic information when it's in that knotted state.
Right, it's just a clump.
Yeah. So when the bacterium needs to replicate or transcribe that DNA, it has to temporarily uncoil it. And that requires these highly specialized enzymes called topoisomerases.
Okay, these are like the little hands trying to untangle the headphones?
Basically, yes. The bacteria use DNA gyrase, which is topoisomerase the key fee, to manage all that physical tension.
And how do they do that?
Well, DNA gyrase operates right ahead of the replication fork. It literally cuts both strands of the DNA, right? It physically snaps them. Then it passes another loop of the chromosome through that break to relieve the tension and perfectly reseals the cut.
Nip. Pass through, reseal.
Exactly. And topoisomerase 4V does a similar cutting and resealing thing, but its main job is separating the interlinked daughter chromosomes after replication is totally done.
Okay, so where do the fluoroquinolones come in?
They act directly on that untangling process. So they enter the bacteria through these water-filled channels in the membrane porins, and they bind to both the enzyme and the DNA at the exact moment the strand is cut.
Oh, so it's like pouring super glue right on those hands while they're holding the cut wire.
That is exactly what happens. The drug forms this rigid ternary complex. The enzymes made the cut, right?
Yeah.
But the fluoroquinolone physically blocks the resealing step.
So the DNA is just left broken.
Yeah. The replication machinery basically crashes into this frozen roadblock, causing lethal double-strand DNA breaks. The bacterial cell effectively shreds its own genetic code.
And just dies.
Very quickly, yes.
That's incredible. And, the evolution of these drugs in the notes is fascinating, too. You can really see how chemists physically altered the molecules over time to get better results.
Oh, yeah. The generations are very distinct.
Like the first generation, right? Nalidixic acid. The notes say it was pretty limited.
Yeah, very limited. It couldn't achieve high systemic concentrations in the body, so it was mostly just used for uncomplicated urinary tract infections.
And then they tweaked it.
Right. Chemists realized, hey, if we add a fluorine atom to the central ring structure, we can dramatically alter the pharmacokinetic profile.
Hence the fluoro in fluoroquinolones.
You got it. That fluorine atom made the drug more lipophilic. It allowed it to penetrate human tissues deeply and cross those bacterial cell membranes much more efficiently.
And didn't it also make it bind harder to the enzymes?
It significantly increased the binding affinity, yeah.
So that single tweak brings us to the second generation, and that features ciprofloxacin.
Super right, the heavy hitter.
Yeah, Cipra is a massive step up. By adding a piperazine ring to the molecule, it suddenly gained this incredibly potent activity against gram-negative bacteria.
Which is huge.
It became a frontline defense against things like Pseudomonas aeruginosa, which is awful for cystic fibrosis patients, and even anthrax.
Yes, Cipro's a serious weapon, and the innovation didn't stop there. We moved into the third and fourth generations with drugs like levofloxacin and moxifloxacin.
How are those different?
Well, while Cipro mainly targets the DNA gyrase in gram-negative bacteria, these newer ones were modified, so they also strongly inhibit topoisomerase the Phe.
Oh, so they're hitting both enzymes hard.
Exactly. That dual targeting expanded their coverage to gram-positive organisms like Streptococcus pneumonia and even anaerobic bacteria without losing that gram-negative power.
Okay, so we have these incredibly sophisticated, deep-penetrating molecules. But looking at the clinical data here, there is a massive practical hurdle.
You're talking about absorption.
Yeah, just getting them into the patient's bloodstream. Because
Yeah.
the notes highlight this wild interaction with dietary calcium, magnesium, and aluminum.
It's a huge issue in practice.
Like, if I take ciprofloxacin alongside a simple glass of milk or a yogurt or an antacid, the drug's absorption just completely plummets. Wait, so eating breakfast can literally stop my antibiotic from working?
It absolutely can. And it all comes down to a chemical process called chelation.
Chelation.
Okay.
Yeah.
So dairy products are packed with dietary calcium. Antacids rely on aluminum or magnesium. Multivitamins have iron or zinc.
Right.
These are what we call divalent or trivalent allocations. They're metal ions with specific positive charges.
Okay, so what happens when they meet the drug?
Well, when an orally administered fluoroquinolone bumps into these metal ions of the GI tract, the drug molecule physically binds to the metal.
They lock together.
They lock together and they form this bulky, insoluble complex.
And because it's so bulky,
Exactly. That new complex is physically too large to pass through the intestinal wall, so it completely bypasses the bloodstream.
wow. So the patient thinks they're treating this severe systemic infection, but the antibiotic is just passing straight through their digestive tract.
Straight through to the toilet.
It is striking to think that, like, a weapon designed to treat anthrax can be completely neutralized by a bowl of cereal.
It really is. That's why pharmacists heavily emphasize spacing out these medications from dairy and vitamins by several hours.
Right. But even beyond absorption, there are warnings about what happens once the drug does reach the systemic circulation.
Oh, the side effects are significant.
Yeah, the FDA's black box warning for tendinitis and Achilles tendon rupture, that was a prime example for me of just the unpredictable nature of systemic pharmacology.
Yeah, it's a very scary adverse effect.
The mechanism really caught my attention because we have a drug designed to target bacterial DNA enzymes, right? But it turns out it can also upregulate these enzymes called matrix metalloproteinases in human tissue.
Yes.
And those are enzymes that literally degrade collagen. So the drug is actively weakening the structural integrity of human connective tissue.
Specifically in load-bearing areas, yeah, like the Achilles tendon. The risk of this cartilage erosion is severe enough that the warning explicitly calls out older patients, people who've had solid organ transplants, and anyone taking corticosteroid therapy.
Because corticosteroids already compromise tendon strength?
Exactly. That combination is a recipe for a SNAP tendon.
The notes also detail phototoxicity.
Right, severe sunburns.
Yeah, the drug circulates into the skin, you get exposed to UV light, and boom, severe reaction. And it can even cross the blood-brain barrier.
Yeah.
It can. It displaces a neurotransmitter called GABA from its receptors in the brain.
Which does what?
It leads to central nervous system overstimulation. So dizziness, headaches, and in really rare cases, actual seizures.
Geez. So we really see how targeting the physical structure of bacterial DNA carries all these systemic risks for the patient.
Which is why there's a completely different approach.
Right. Stepping back from the DNA itself and targeting the metabolic assembly line that actually builds it.
Yes, that brings us to the folic acid antagonists.
And the strategy here is basically starvation, right?
Exactly. Both humans and bacteria require folate folic acid to survive.
Right.
It's this critical coenzyme used to synthesize purines and pyrimidines, which are the foundational building blocks of RNA and DNA.
Mhm.
Without folate, a cell simply cannot synthesize genetic material.
No genetic material, no division, they can't multiply.
Right. But the biological exploit, the vulnerability we target, lies in how we obtain that folate.
Okay, because mammalian biology, we just absorb preformed folate from our diet, right? Like eating leafy greens, spinach, fortified grain.
Yes, we eat it. Bacteria, on the other hand, lack the transport proteins necessary to absorb folate from their environment.
So they're completely impermeable to it.
Totally impermeable. To survive, they are forced to synthesize their active folate entirely from scratch, inside their own cell.
De novo synthesis.
Exactly.
Yeah.
And they use a precursor molecule called para-aminobenzoic acid. PIA.
Okay, PIBA.
Yeah.
I view this internal bacterial synthesis like an automobile assembly line.
Okay, I like that.
So you start with the raw material, the PIBA, and that PIBA gets fed into the first major machine on the line, an enzyme called dihydroxyapatite synthetase.
Right.
And that machine stamps out this intermediate product, like the car chassis. Then that chassis rolls down the line to a second machine, an enzyme called dihydrofolate reductase, which puts the wheels on and refines it into the final active folate molecule.
That's a great way to look at it. And the first group of drugs designed to wreck that factory floor are the sulfonamides.
Sulfa drugs.
The sulfa drugs, yeah. They are structural analogs of PIBA. They basically mimic the chemical shape of the raw material almost perfectly.
Oh, so they act as chemical decoys.
Exactly. Competitive inhibition. That first bacterial enzyme, dihydroxyapatite synthetase, grabs onto the sulfa drug instead of the real PIBA.
The machine jams.
The machine jams, the assembly line stops, and the bacteria can't produce that intermediate chassis.
But to guarantee the pathway is completely shut down, the notes say we introduce a second drug, trimethoprim.
Yes, the 1-2 punch.
Right. Trimethoprim targets the second machine on the assembly line, the dihydrofolate reductase, so it binds to that enzyme and stops the chassis from becoming the final active folate.
And this is key. Using either of those drugs alone is just bacteriostatic. It stops the bacteria from growing, but it doesn't necessarily kill them outright.
But when you combine them...
When you combine them, it creates a synergistic blockade. The combination drug is called cotrimoxazole, which pairs sulfamethoxazole with trimethoprim.
And by jamming two sequential steps in the exact same pathway, it becomes highly bactericidal. It kills them.
It's incredibly effective. Cotrimoxazole is a vital tool. We use it heavily for community-acquired MRSA, which is, highly resistant to standard penicillins.
Right.
It's also the primary treatment for pneumocystis jirovecii pneumonia.
Oh, the fungal infection.
Yeah, that severe opportunistic infection that targets immunocompromised patients, especially those with HIV or AIDS.
Okay, so this selective toxicity seems perfect on paper. Humans don't synthesize folate from PAPA. We don't even have that first machine, right?
Right. No dihydroxyapatite synthetase in humans.
But wait, if humans absorb folate from food, we still have to process it. Don't we have our own human version of that second machine, the dihydrofolate reductase?
We do. We absolutely do. And that's where things get a little tricky.
Ah.
Trimethoprim has an affinity for the bacterial version of that enzyme that is roughly 50,000 times greater than its affinity for the human version.
50,000 times, okay.
So it vastly prefers the bacterial target. However, it's not perfect. In patients receiving really prolonged therapy, or high doses, or folks who already have a folate deficiency.
Methoprim starts jamming the human machines.
Yeah. It can interfere with human folate metabolism, and that disrupts red blood cell formation, leading to a condition called megaloblastic anemia.
But fortunately, the notes say there is a pharmacological workaround for this, right?
There is folinic acid.
Okay, not folic, but folinic acid.
Right. Patients experiencing this anemia can be given folinic acid. It basically provides the already active form of folate directly to the human cells, completely bypassing the drug's blockade.
Oh, that's clever. And because bacteria can't absorb outside folate at all, giving the patient folinic acid doesn't accidentally rescue the infection?
Exactly. The bacteria still starve.
Wow. But what about the sulfa component? Because sulfa carries its own set of weird complications, right?
Yes, physical and metabolic complications. So sulfonamides are metabolized in the liver, primarily through a process called acetylation.
Okay.
And those resulting metabolites have to be excreted through the kidneys. The problem is, in neutral or acidic urine, these acetylated metabolites have very low solubility.
Meaning they don't stay dissolved.
Right. They actually crash out of the solution and precipitate into solid physical crystals right there inside the urinary tract.
Crystalluria.
Yes.
Just thinking about developing physical crystals inside the kidneys or the bladder.
Right.
That leads to severe nephrotoxicity, right? Tissue damage.
It's extremely painful and damaging. And it reinforces why aggressive hydration is a mandatory clinical instruction for anyone on a sulfa drug.
You have to drink a ton of water.
Tons of water. Keeping the urine highly dilute prevents the concentration of the drug from ever reaching that precipitation threshold.
Makes total sense. And beyond the kidneys, the notes highlight a really critical danger during pregnancy and infancy with these drugs.
Yes, this has to do with plasma proteins. So in the human bloodstream, you have bilirubin.
Which is a natural byproduct, right? Like from old red blood cells breaking down.
Exactly. Normally, that bilirubin is bound safely to serum albumin in the blood, so it can be transported to the liver and processed out.
Safely tucked away.
But sulfonamides have a really high affinity for those exact same plasma proteins. They aggressively compete for those binding sites on the serum albumin.
Oh, I see. So the sulfa drugs just outcompete the bilirubin. They forcefully displace it, essentially kicking it off the transport proteins, leaving all this free-floating bilirubin in the blood.
Right. And in an adult, that's one thing. But in a newborn, the blood-brain barrier is practically non-existent. It's highly permeable.
Oh, no.
So that free bilirubin isn't just circulating safely, it crosses straight into the baby's central nervous system.
Leading to kernicterus.
Yes, kernicterus. It causes severe, permanent neurological damage. Brain damage.
Which is why sulfonamides are strictly contraindicated in pregnant women near term and in infants under 2 months old.
You absolutely cannot use them in that population.
Okay, so we've seen how these systemic drugs, whether they are shredding DNA structure or starving metabolic pathways, they circulate everywhere and they carry these massive risks of systemic collateral damage, right?
Yeah, tendon ruptures, crystals, neurotoxicity.
But what if a patient just has a localized, uncomplicated infection, say just in the bladder? Sending a heavy-hitting drug through the entire vascular system feels like a massive overreaction.
It is. You don't want to expose the whole body to side effects if you don't have to. You need a localized weapon.
And that brings us to the urinary tract antiseptics, specifically methenamine and nitrofurantoin.
Right. The pharmacokinetic profile of these drugs is entirely different from everything else we've talked about.
How so?
When you administer them orally, they are absorbed, but they're excreted so rapidly by the kidneys that they basically never achieve a therapeutic antibacterial concentration in the blood plasma.
Oh, so if a patient has pneumonia or like a blood infection, these drugs are completely useless.
Entirely useless. They bypass the broader body and deliver their payload exclusively where they concentrate, which is in the urine.
Okay, so how do they kill the bacteria once they get to the bladder?
Well, nitrofurantoin is reduced by bacterial enzymes into these highly reactive intermediates that basically attack multiple targets at once. Ribosomal proteins, DNA, cellular respiration.
Just a shotgun blast.
Pretty much. But methenamine relies on a purely chemical trigger based on the environmental pH of the bladder.
This was the most mind-bending fact in all the sources for me, the formaldehyde trick.
It's incredible, right?
Yeah, because methenamine is just a precursor molecule. It has zero antimicrobial properties on its own.
Nothing.
But when it enters an environment with a pH of 5.5 or lower, it rapidly hydrolyzes. It breaks apart into ammonia and formaldehyde.
Literal embalming fluid.
Yes, the drug uses the acidic environment of the human bladder as a chemical catalyst to synthesize formaldehyde right at the site of the infection.
Directly in the bladder. And formaldehyde is a incredibly potent, broad-spectrum chemical cross-linking agent. It denatures the proteins and nucleic acids of almost all bacteria on contact.
And the best part is, because the attack is so fundamentally destructive at a basic chemical level, bacteria have no enzymatic workarounds for it.
Right, they cannot mutate a structural protein or alter a binding site to survive embalming fluid.
Resistance to formaldehyde just does not develop.
But, and this is a big, but to spring that chemical trap, the urine absolutely must hit that critical pH threshold of 5.5.
Right, it has to be acidic, so clinicians use dietary interventions, right?
They do. Patients on methenamine are frequently told to drink lots of cranberry juice or take high doses of ascorbic acid vitamin C.
Which naturally acidifies the urine.
Exactly. It lowers the pH, guaranteeing the methenamine will rapidly convert into its active formaldehyde state once it pools in the bladder.
It's just brilliant chemical engineering. You're intentionally using your diet to manipulate your internal pH, triggering a localized chemical reaction that obliterates an infection, all without exposing your systemic circulation to the drug.
Though we should point out, the sources are very clear on this. You absolutely cannot mix these local urinary antiseptics with the systemic sulfa drugs we discussed earlier.
What?
Because if formaldehyde is present in the urine alongside sulfonamides, the two molecules chemically react with each other.
Oh, no.
Yeah, they form an entirely new, highly insoluble compound. The resulting precipitation massively increases the risk of severe crystalluria.
So even worse crystals.
Much worse.
Wow, it is all so highly interconnected. Like pulling all these distinct pharmacological strategies together really reveals the deep, deep complexities of selective toxicity.
It really does.
I mean, we've explored how fluoroquinolones exploit the unique supercoiling tension of bacterial DNA, basically dropping superglue on those topoisomerase enzymes.
Yep.
We've seen how folic acid antagonists use competitive decoys to starve bacteria of their metabolic building blocks. And finally, how urinary tract antiseptics literally use the physical pH of the human bladder to unleash localized formaldehyde.
And of course, along with all the immense physiological rules governing their use,
Right.
the necessity of avoiding calcium in milk with fluoroquinolones to prevent chelation, the critical need for aggressive hydration with sulfonamides to prevent kidney crystals, and using vitamin C to catalyze methenamine.
Every single chemical intervention carries a specific requirement to ensure it actually reaches its target without taking out the host in the process. We really have built this incredible arsenal of precision tools based on observing the microscopic differences between our biology and bacterial biology.
We have. But the underlying reality here, the thing you have to remember, is that selective toxicity relies on targeting a static biological structure.
Okay.
And bacterial biology is anything but static. The sources we've analyzed today constantly reference the rapid development of resistance.
Right. They fight back.
Constantly. Bacteria are continuously selecting for chromosomal mutations that alter the very topoisomerase enzymes those fluoroquinolones are designed to target.
They just changed the locks.
Exactly. They undergo plasmid-mediated exchanges to acquire new resistant forms of dihydroapatite synthetase to completely bypass the sulfa drugs. They even construct microscopic efflux pumps within their cell walls to physically pump these antibiotic molecules back out the moment they cross the membrane.
So the moment we engineer a new generation of drugs, they just evolve a new defense.
Exactly. Which leaves us with a really critical, lingering thought for you to ponder. Are we actually achieving permanent victories in our understanding of pharmacological precision? Or are we merely running on an evolutionary treadmill, constantly forcing pathogenic organisms to mutate into forms that our most sophisticated chemical traps can no longer catch?
Man, that is the defining challenge of modern medicine right there. Well, thank you for joining us on this deep dive into the pharmacology of antibacterial agents. Keep questioning the mechanics of the world around you, and we'll be here next time to help you look a little closer at the invisible systems keeping us moving.